All right, we'll go ahead and get started. Hello, and thank you for joining us today. My name is Michaela McShane, and it is my privilege to welcome you to the Startup NASA Feature Series, hosted by NASA's Technology Transfer Expansion Program. T2X expands the tech transfer mission of ensuring innovations developed for exploration and discovery are broadly available to the public, and include a focus on entrepreneurial innovation and strategic partnerships to help accelerate commercialization. This monthly series is an opportunity for NASA technology-inspired startups to share their experience. This month, we'll be hearing from Agscent. Please note that your microphones will be muted throughout this presentation, so if you have questions, please share them in the chat box in the lower right-hand corner of your screen. We will answer questions at the end. Thank you for joining us today. All right, it is my pleasure to introduce today's speakers, Dr. Bronwyn Darlington and Ashley Sweeting, founder and vice president of Agscent. Dr. Darlington has taken NASA technology designed for use in space missions and transformed it to be able to detect pregnancy and identify disease in cows. Her understanding of the needs of primary producers, dairy, grazing, and feedlotting is born from living and working on the family's 5,000-acre cattle and sheep property in Australia. Driven by the need for advanced cattle management techniques and a deep knowledge of the way technology in other sectors has disrupted and transformed them, Bronwyn founded Agscent. She sought to adapt breath diagnostics technology designed for humans and apply this to pregnancy detection and disease identification and management to transform animal health and care. This concept gained momentum with the support of NASA and their e-nose sensors, which have now been commercialized and are about to transform the way livestock live and breathe. Bronwyn brings more than 30 years of industry experience in a variety of sectors, including senior roles in mining, engineering, agriculture, local government, finance, textile and apparel design manufacturing to bear in Agscent. She has a PhD in economics focused on consumer psychology and behavioral economics. It is her work teaching the Sydney University Global Executive MBA subject, Finding Opportunity and Disruption, which linked technology developments in space, medicine, and AI to the opportunity available for livestock and precision agriculture. She is a serial entrepreneur and inventor. Ashley Sweeting specializes in commercializing cutting-edge biotechnology for animals and leads the NASA Technology Transfer Program for Agscent. This role includes engaging and developing partnerships across the livestock industries, the research community, investors, and manufacturers, alongside working with NASA scientists on the technology transfer. He has worked on cellular health, the microbiome, and building sensor technologies. Previously, Ashley worked in Asia, the Middle East, Africa, and Europe on programs to improve agricultural productivity and sustainability, combating famine. He holds an MBA from the University of Tasmania, a Bachelor of Science in Agriculture from Sydney University, and a graduate certificate in data analytics from Boston University. And with that, I will hand it over to our presenters to tell you a little bit about their company.
Alrighty, thanks, Ash.
Thank you very much.
All right. Okay, so thank you so much for coming online today, and I'm really so excited about sharing where we're up to and answering the question I think that is probably the most commonly asked: what does NASA care so much about cows? So I'm going to sort of link those pieces together and help you ideally understand that what we're actually doing is we're really a biotech company that's taking technologies from many sectors and putting them together in a way that is solving a real and significant global problem, and that is the way that we manage livestock and that what we need to do in the future. So the first thing I wanted to do was, though, thank a few people who've been really key and very important, particularly in the NASA relationship along the way, and that's the staff at NASA Ames. When I first was sitting on our family farm thinking, oh, where is all the money for this? Where is the best sensors? Where is the best technology? And I thought, you know what? I'm going to email NASA. I never thought that I would meet such a wonderful group of people that have been so supportive. And I just particularly wanted to give a couple of shout outs. One is to Kim Hines, Kim Manifra, and Nari Ahn, who have really been the tech transfer office so helpful on every level. But also equally important, actually, you know, that everyone is important, but Dr. Jing Li, who's invented this technology, and her assistant, Ami Hanon, have really worked so closely with us. And I've learned an awful lot thanks to these extraordinary people. I just want to thank my own staff and those who've helped us on the way, and I will pull those out as we go along. But the first thing I want to do is sort of run through a bit of a presentation and try and give you a flavor of what is Agscent actually about And why do we need NASA technology? So I'm just going to share my screen, and we have practiced this, so I'm hoping this will work as sweetly as it could. Here we go.
Looks good.
Thank you. Right. So in my mind, I'm coming from a primary producer background, but at the same time having a very strong future technology orientation and work in industry and global industry at that. Bringing these two technologies together, it gives us an opportunity to transform the way we think about molecules in air, and for me in particular, about molecules in livestock breath. So for us, the future needs to be measured. What we need to do as we go further forward in our use and working with animals is to ensure things are measured, sustainable, and we really are ready to take off. So imagine this if you could, that anyone could breathalyze a cow and identify pregnancy, disease-related biomarkers, or measure methane using nanosensors in a way that is easy to use, non-invasive, efficient, accurate, early, affordable, and at the point of care. This is the pursuit that we have in being able to produce food with livestock, but also to ensure that what we're doing is to support a sector that is very important to our economies, but do it in a way that at the point of care, which improves animal welfare and the human welfare as well. But where are we actually from, and why Australia? How does that fit into the picture? I'm just going to play a short clip that just anchors me and where our farm is in New South Wales and helps you understand how many people, what is involved in going from one part of the world to another with a technology like this. Will come up in a sec.
Agscent is a company that is inventing breath diagnostic technologies for livestock. The Agscent breath diagnostic device is able to be placed at the cow's nose and within a few seconds take enough breath to be analysed by sensors and give you a diagnostic for pregnancy at day 18 after insemination. It takes about 60 seconds all up and you have the answer in your hand. We've been able to grow our team to include so many ages and individuals with different technical expertise. And being in New South Wales, I have to say, has been a great opportunity to pull from many areas of our ecosystem, from science and education, to bring them into our team. Agriculture and livestock is really moving towards precision and data-driven decision-making. The livestock and agricultural sector is being driven forward by its need to be more productive. We've developed this product to feed into the data stream to allow producers to have accurate, affordable, and immediately accessible information about their animal health, welfare, and their health status. Being in New South Wales is a great opportunity for us. We have access to great researchers, we have fabulous universities, but the New South Wales government has actually opened the doors to the world for us. We have a relationship with NASA that has been really fostered by the New South Wales government in ensuring that we have the right structures and setups internationally, both for future export, but also for research collaboration. We're solving a global problem with export potential all over the world.
So where we come from has been really important to where we're going. And I wanted to really focus on the problems that we're solving and how the technology is able to do that. And I'd really encourage anyone, if you come up with a question, just to throw that into the chat so that we can answer those later on. But why start with livestock reproduction management? One, that we are a cattle breeding family, and that's the core aspect of business. But what it really is, it's the reproduction management is the key to beef and dairy profitability. And I'm a real fan of starting where the business need is. Identifying and being able to manage animals with reproductive problems, which is approximately 10% of each herd, optimizes herd productivity. So if you're going to get the attention of potential users, we know we need to speak to profitability and productivity. By ensuring only productive animals are in the landscape, this reduces environmental impact, which is critically important and part of everything that we do. Knowing herd reproduction status allows better management of feed costs, pasture management, animal management, and maximizes future income. But in the time of climate change and really understanding the role of livestock in this, in the recipe that is CO2 and methane in the atmosphere, we've come to realize that low reproduction rates, which are prevalent in some cattle systems, result in higher methane emissions per kilo of beef. So by increasing calving rates, which means removing non-productive animals, this decreases methane emissions per kilo of beef. Sorry. So how does it actually work? I'm going to explain it, then I'm going to demonstrate it for you. The reality is that the way we all deal with animals when we place them into a safe environment to be able to work with them doesn't change with what we're doing. But from a preg testing point of view, and close your eyes if you guys aren't into this, I'm just going to explain how it currently sort of works. Everything comes from the back end, and it's not, it's definitely one of those things that if I just mimic it, we put a rubber glove on and I won't go further. So we need to do things differently. And this is a real motivation for me standing in the cattle yards preg testing animals with our vet the same way we have since the pyramid's going. This is just, we have to do things differently. So the cow is secured. Then there is we've developed this thing called a breath diverter. As you can imagine, and I'm sure you guys, COVID's changed everyone's understanding of breath. Before COVID, I think we all sort of thought that we breathe in and we breathe out, and we really think about it only when we're doing meditation or maybe yoga. The reality is it's extraordinarily rich biosource. And so what we needed to do, because we work with cattle in the most extreme environments, so we've been collecting over 5,500 cattle sample breath samples now, and it's been snowing, it's been dusty, it's been beautiful and sunny, it's been incredibly rainy, there's been pollen in the air. So I had to invent a technology that's able to actually open and close and collect only the very narrowest of end tidal breath. Anything that is just this last key part of tidal breath. That's all useful, except you need to be able to do the diagnostics at point of care. And Ash is going to explain a lot more about the eNose itself. The next step is that we plug the small capsule into the eNose, and then it's able to process that over the NASA sensors and give a diagnostic of pregnant or empty, and which it means open or not actually pregnant. But I'm just going to stop the sharing for a second. I'm going to give you a little demonstration, because this is the breath diverter device that we have. So this is taken, and I'll show you the pictures in a minute of the development of it. So many iterations, because cows, you know, when I first started this, I thought, okay, there's going to be a lot known to science. We're going to understand cow breath. And we didn't. We understand that cows all probably breathe like humans, right? Just breathe out. Every cow breathes differently. And then we'd understand a lot of the biological processes. Well, we didn't either. So we've had to do a lot of work with all of those areas. But in its simplest form, this is what we have. This is the latest technology. So if I turn it on, it makes a whole pile of noises, has some flashing lights, because we all love flashing lights. And then it tells me that it's ready to scan the animal tag. So what this does is you're able to put this at the ear tag. In Australia, they're regulated. In many industries all over the world, that's increasing, but allows you to do the identification. By pressing the next button, it says, if you can see that, it says collect breath. At the moment, and we're always evolving, because this is a first of all technology, we have this little bag. This little bag is all the breath that you need. In fact, you only need half of this. And what I'm going to do is I'm going to breathe. It takes 2 breaths to calibrate to any animal in all reality, to get the end tidal at the 3rd breath. So there we go. So this little capsule, which we will make really fun eventually, is then plugged into, reaching out a camera. I should never do that in a presentation. Anyways, plugged into the eNose device. With a cow, it takes approximately 10 seconds to get the breath, and it takes 10 seconds to run it over the eNose sensor. Then the diagnostics are communicated, and we're at this point in time in the product development when we're refining the algorithms to be able to manage all the environmental variability that exists, which is huge, and the biological variability across animals. So I'm going to pop back into the share screen again and explain that we're also now beginning to look at disease. So if you think about this little bag of breath, if you can still see me, what is in here, we started to analyze. So the way we do the analysis is to take the exact same methods of analysis of molecules in breath that we do with humans, particularly for cancer diagnostics. So because there wasn't a lot known in science at the beginning, what we started doing is working with GCMS and taking the exact same protocols that you would work with in any kind of breath diagnostics, and putting the breath through gas chromatology mass spectra to be able to identify what molecules are there. Because pregnancy is actually an extremely complex phenomena, and it's the up and down regulation of over 100 molecules at the same time, which is why this has not been possible until AI has developed to the level that it has to be able to use machine learning, and we use neural networks to be able to create algorithms that can manage the variability. In any breath, there's over 3,500 molecules. So once we started decoding breath for pregnancy, we began to see other molecules in the same bag of breath. They were bigger bags of breath at the beginning. Some of those related very particularly to certain diseases. So in the cattle world, again, because we started with there, though you'll see in a couple of minutes with some photos of sheep and other animals that we've been looking at more closely now, bovine respiratory disease, Campylobacter fetus and pestivirus are common and very costly diseases. BRD is complex. It's 5 bacteria and 4 viruses that all actually work together in different ways to make an animal very sick. Currently, there is an inability to rapidly detect many diseases, and this increases transference, reduces animal welfare and condition, and increases the cost of management. And billions are spent across the world to treat and prevent and treat diseases within the global cattle industry. Biosecurity is increasingly important. And we know that with the continuous movement of animals across borders, the great risks to our own ability for food production. So once we started looking at the idea of how, so we can map and what we call create molecular blueprints for pregnancy, we thought, let's start looking at other diseases. And because I'm a real fan of leapfrogging, so let's not just look at the one key component that we can move to, but let's go further out and look back at what we could achieve, knowing what other technologies may exist at the time. We thought that the key issue here may be to separate out the bacteria from the virus contributors to bovine respiratory disease, because when it comes down to the NASA census, they were actually designed for human disease. So I thought, well, let's look and see what we can do here. So the short story of disease is that we've been able to map the molecules that come particularly from each 5 bacteria and each 4 virus separately. Now, some of these viruses are very particular to cattle, but some of the molecular constructs in these viruses and the bacteria actually do cross over into human lung diseases and lung infections, both bacterially and virally. So we're now developing the pipeline of products that we will then be able to bring out, so that my goal is that you will have a universal source of data about the biological status of animals. And to do that, we're broadening. We do work with NASA, but we're also working with other universities as well. But again, once you begin to see what's in the little bag of breath, we actually start to see methanogenic molecules. And we are in a race to methane zero. Yes, we're in a race to carbon zero, full stop. But methane is really the safety cord that we have that we can pull if we can reduce methane, because it has a shorter life in the environment. So with the climate crisis that we all know we're in, what we wanted to do is say, well, since we're really quite good now at being able to collect breath from animals, let's look at how we can create new solutions that can be at the point of care to support this journey to methane zero. So we've put together technologies that's slightly different sensing technology at the moment, but we're spending the time over this next 12 months bringing together the nanotechnologies that are with NASA and others that we also are creating to be able to solve this problem. But where did we come from, and why, again, why the question of why is NASA interested in cows? Well, back in 2017, when I was in the yards preg testing, the top left-hand corner of my screen is Bill, our vet, preg testing the cattle. I'm like, come on, this has got to change. We need to do things differently. But I wasn't really sure where to even get a sample from. So on the left-hand corner, there's bags of breath that we created, blood samples, urine sample, everything you could possibly say. And I do get asked by people quite regularly, why don't you just take urine? So if you know anything about cows, you'll know that they're very big creatures. And I do joke about the fact that you would actually need an umbrella, and you'd need to be a long way away, because once they start to give you a urine sample, it's a really messy thing. In addition to that, a whole lot of the other girls who will be in the race will also decide that now must be the time to do the same thing. And so collecting samples is really tricky. Breath is a universal sample that ultimately we really will roboticize and take the human out. But you can see we've gone from different prototypes of 3D printing things, tubes up noses, cones on noses, through to the 2018 period, of really developing the method and building our team. So we started having an awful lot of fun, but you'll see everything is out in the field. And this is because I really wanted to work with producers, because if it's a problem that they want solved and they're going to engage in, then there'll be a market for the product at the end. And you can see on the right, that's Darren, who's our COO, going from really struggling with animals to being a very simple system. That was the glass jar model, which we moved further and further from. But we also started to play around with nanosensors. But this is where we really struggled with the consistency of nanosensors. In 2020, we tried various versions again and back out in the field. We spent a lot of time in the field. So one of the reasons I can answer the question of why is NASA interested in cattle, I'm not particularly interested in cattle, but what they are actually interested in is American industry and ability to provide food, and this key point of leveraging the technology for one sector being space and seeing it productive elsewhere. But we had a really great advantage. Because we had 1,000 cattle at home, I was able to continually bring cattle into the yards to do mass sampling, and that's allowed us to develop the technology more rapidly. We have even more sheep, and we're connected to many others as well. So 2021, we're out again, and then we started to play around with sheep, because curiosity is, well, what about sheep? Maybe, what about other species? What's the biological similarities? What are the differences, and how can we collect breath from other animals and analyze that the same way? So we've created a methodology of collection, methodology of analysis, the ability to map molecular blueprints, and then now we are not functionalizing sensors to be able to read those real time. And we get to have a great deal of fun out in the field. But in 2021, this is where I was sitting at home going in the middle of COVID, going, what are we going to do? Where is the money? And I did literally go through the NASA patent library to look to see whether any of the breath technologies that NASA was developing could be transferable. And I found molecules that were identified in the patents that I was seeing in cow breath. So I really did send an email inquiry to NASA, to the NASA inquiry number for tech transfer. And within 24 hours, it actually had a response. One, saying that's extremely novel, but two, being able to help me with the pathway of who I need to talk to, how I need to do this, to be able to explore whether this would work. And this is where I started to meet the most amazing people in NASA Ames. And again, just thank you for everyone's absolute support and engagement. But the funny thing was, what I ended up doing was jumping on a plane and coming over to California. And the top is one of the big dairies that's up at Modesto, driving up to Modesto, collecting bags of breath, which is in the top right corner, which is the back of my car, driving that down to NASA Ames. And I still do not know what the security guys think by me parking the car park, and I would open the back of my car, take out bags of breath, and then I'd give them to Jing, who is in the bottom screen on the left too, or Ami, who assists her in the laboratory, and she would take the bags of breath into NASA and then run them over the same sensors that they were doing for COVID, and the same sensors that Simplify is commercializing, who was last week's call, for humans. So I did that several times to be able to have the confidence that what were results we were getting were potentially valuable and that we could work together. So that took a while. We went out again and out again and out again. And in 2022, we did a whole pile of trials both in Australia and here in the US, just to validate both the science, the breath collection, and our ability to use sensors. But then in December 22, Ash Sweeting joined me, and we had by then actually signed off on the commercial license for the technology and begun to work very closely with both Jing and me, who have been wonderful, to be able to help develop the technology itself. I'm going to pass to Ash now to explain how that works.
Thank you, Bronwyn. So I'm the very fortunate person who's been heading into NASA Ames most weeks over the last 14 or 15 months to work with Jing and me, the two people in the background in that picture, to actually take the NASA technology and what's called knowledge transfer. So the way the US government works, excuse me, the government can't give us anything, but they have to teach us how to do it. So working with Jing and me have been working on this for 10, 15 years. I had to take everything they'd learned in that time and, you know, cram that into a 1-year time period. So the first part of that was actually the technology, the sensors and the doping materials that they had made, and I had to replicate that. So go and buy the ingredients for that and then learn how to make those chemicals. And then finally, the harder part, well, from my perspective, the harder part was actually put those molecules on a chip. So I am just going to stop sharing, and I have a small video that I will share with you as well. And this is the process of actually doping the chips. I'll just talk through it while it's going. So you have these materials that are in that little vial, and you have to take a 0.01 microliter drop of that material, which is a mixture of various carbon nanotubes that are functionalized with different compounds and different elements, and put that 0.1 microliter drop onto one of those little tiny circles. And that's something that takes quite a bit of dexterity and quite a little bit of practice. So Jing and me, there was a lot of patience working with them in terms of the first versions, which were a big mess of everything mixed together, to finally make it something that was usable. And then the next stage was to actually go and test that. So we had to have our own device. We had to then insert our own chips into our own device. And then that was back out in the field, working with farmers and ranchers to collect breast samples, once again, taking them back into the NASA lab and running through and analyzing those samples, going through, then having to adjust the substances, adjust and fine tune and optimize the different chips, and working through that process iteratively to actually get the accuracy that we want. The other thing, what we've done as well, is we've migrated from the square or the rectangular NASA chips that you can see there to, if you can see that, these round ones. So that's the next version of the technology which we're now working on. And that video is ended, so I will stop sharing that and go back. And we're now in the stage where we're collecting thousands and thousands of samples and processing them across this the eNose sensors to develop and fine tune the algorithm. And back to this, these chips will be a bigger screen that you can see them on. They're now in the second generation of the prototype, which is the one that we have worked with Essential up in Oregon, in Corvallis, to develop this. Thank you very much. Exactly. They've been great to work with. And that's the second generation of this prototype. And this is the only one available currently. There's some more that will be with us in the not too distant future. But once again, it's the same. It's that NASA smarts, and it's the, I guess, decade plus of work that they have put into developing the technology, working alongside me and talking with her. You know, she's explained that just one of those doping substances that, you know, we've had access to through this program has taken, it's been a single PhD just to develop one substance. So there's a huge amount of work that's gone into that. And I think one of the things I've really loved about working with NASA is when they get, they don't shy when there's a big problem. They keep on working on it, keep on working, find the solution, work through all those problems. And it's that kind of persistence and that determination which has been an absolute joy to work with.
Brilliant. Okay. Thanks, Ash. See, it's so much fun to learn something and do something new. When I asked Ash if he'd like to come work for Agscent, I don't think there was any way I could have explained exactly what that was going to entail and the new skill set that was going to be necessary. But I think that's the thing about inventing new technologies and trying to solve problems that aren't traditional sources of this kind of technology. We all have to stretch and think about this differently. So it's been a great part of the journey to do so. So just going to ping back to the presentation part and come back into here. Just to wrap up with where we're going to go from here. As you can see, I've tried to explain that once you have one breath source and are able to replicate end tidal breath, what that does is opens the doors to lots of opportunities and lots of future possibilities. And one of those is robotics. The ability to move from sensors where Ash has to do the hand doping to being able to get reproducibility is one of the major challenges of going from a one-off product that NASA creates to a fully commercializable product that has the reproducibility that's necessary, so that every time anyone does the same task, you get the same result. So we're at the moment working through that process. It's not the cheapest process in the world, but it's essential to really liberate this technology to be able to be used in so many different ways. To do that effectively, this is where collaboration, and we're big on collaboration, is really important. And so we're working together with Simplify and Optimec and other companies to do that key component. In addition to that, we're sharing resources. Well, they're sharing resources with us at the moment, which has been fantastic with WareCorp, looking at the AI component and understanding how algorithms, I always found algorithms will change the world, but which algorithm on which occasion and how do we actually make that work? So where will this go in the end? This is really platform technology. And being able to make sure that as a founder and as a startup, we stay very focused on proving the technology can work, launching into a market and raising funds for that market is really important for one thing. We've leveraged that for methane because we've been able to put the methane product out to the market really quite rapidly, because it's absolutely critical right now to do so. And we're using various technologies that we are blending in to be able to make the NASA technology even more specific, but also create the robustness that's necessary. Methane emissions are tricky. It's a light little gas and gets blown around by all sorts of things. It comes from eructation. And all those cows, they breathe differently and they all eructate differently as well. So it's been great to be part of that. But what we know is that from a biological point of view, which is a point of where we start, that the findings, what we have is applicable more broadly. And we are looking forward to being able to leverage that. So going to beyond pregnancy to health and disease screening diagnostics, what we're really wanting to do is we're developing this particularly to be able to be smaller and cheaper in any feed trough. And because pregnancy is the up and down regulation of many molecules, it's tricky. You really need end-tidal breath. But with many of the diseases, we're finding the biomarkers are so profound that it's a presence and absence. So we believe that we can do a whole lot more to take the humans away, whether it's in a milking parlour or it's in a feedlot or it's in cattle, to be able to continuously give farmers a point of care diagnostic and integrate that into infrastructure. So I mentioned partnerships. We have a lot because collaboration is really the key to being able to leverage, in my view, to the rest of the world. But because we're dealing with deep science, we're the whole nightmare, right? So we're an industry that doesn't normally do technology particularly well, agriculture. We're deep science, we're deep technology, we're device, manufacturing, robotics, and going to market. So it's a big call. So we need to be able to draw on other expertise, which we're doing. But where are we now? And before we just throw to questions, I just want to put it out. So what are we doing now? As you can see, we've been able to achieve a lot, but we're about to take the next point to scale and the next point to robotic production. So we're raising funds at the moment. We're looking between 5 and 10 million US dollars. The variation is how quickly we would go to other animal species and be able to move to robotics faster. So I would invite anyone who understands, one, the scale of what we're trying to do, and I have plenty of other information about the size of the global market, but also I really do believe that now is the time for precision agriculture to be supercharged by data. And to be able to do that, working with NASA is an absolute breakthrough. So I'd invite anyone to contact either Ash or myself or scan the codes there to find out more about our products. But then perhaps Michaela, I could throw back to you to see if there are any questions from those who are on the call.
Thank you. Excellent presentation. We will now go to questions. So some of these you've already touched on, so maybe just expand a little bit on the subject. Okay, so the first question, and you did already touch on this, but it says, can this technology be utilized for all livestock, or are there biomarkers native to only cattle?
So the question depends on what we're looking at. With certain diseases, there are biomarkers that are very specific to that disease that may actually only occur in cattle or sheep or pigs or camels as it works out. With pregnancy, there is a great deal of crossover. The issue is the opportunity with that is to be able to create different collection techniques. So but we've also created a small vaginal probe, and the reason that's been really amazing is we can take molecules directly from the reproductive tract, and for some animals that is a really great way of removing any ambient or any other contradicting factors. So the answer is there are some applications that will go right across, and there are some that will be specific.
Thanks. Can you elaborate on the safety aspect of the technology compared to traditional methods of pregnancy and disease testing for livestock?
Certainly. So if you're thinking about pregnancy, the most common is rectal palpation. And I've certainly spoken to plenty of vets who in fact, one of the major universities' vet departments said one of our key goals is to stop any vet having to do this as part of their job, because shoulder injuries and injuries to humans are substantial. I've never met a cow that liked this procedure. I've been flicked in the head plenty of times by tails and everything else to show that the dislike of it. For us, the issue is animal handling. These are I can't do the pounds, but I can say that they're 500 kilos plus of please don't be near me. It's risky. So being able to roboticize this is a key aspect of where we're going to go for as far as safety is concerned. Being able to simply take a breath with an animal with its head restrained is very quick, and it is less stressful to the animal, and it's much less stressful and physically demanding on the operator. And ultimately, we'll embed these in equipment and machines.
Great, thank you. What is the sensitivity at, say, day 28 post-breeding? Out of 100 pregnant animals, how many will the sensor detect?
Yes, so I'm going to separate this into two parts. One is the biology of what we're able to do with biology, and that is from day 18, we've reproduced trial after trial after trial of between 92 and 100% accuracy. That's with many different animals. So the science we know is very clear. With the sensors, we're at the point now where we're regularly getting 90%. What we know is with hand-doped sensors, there is irregularity in the doping, and we believe that the robotics is what's going to be able to move us through to increase that sensitivity and specificity, which means get the right number and make sure it's pregnant and empty is correct, pregnant or open. The industry in Australia has set us a target that says that while the average accuracy of pregnancy testing can range from 80% up, the gold standard is 95%. And so that is what we're working towards. For many countries in the world, such as India, they don't actually have any capacity to do this at all. So theirs is just a wait and see model. So we know that even if we were at 80, 90%, that would be substantially an improvement. But with the technology, we believe we'll get there to 95%.
Thank you. Can you speak to the sensor specificity and sensitivity, particularly related to the use of AI for development of molecular biomarkers for health diagnostics and prognostics?
Yes, so the way I try to explain it is the ENO sensors are really like a digital camera. What they see are red and blue dots. AI allows us to model that, to be able to characterize those to mean something. I'm took the view from the very beginning that if we didn't understand what the camera was looking at, what the sensors were looking at, we would misattribute the result. And so in early days, it was honestly, it felt a little bit like a random number generator with some of the sensors that we were using. On some days it'd be great, and other days it wouldn't be great. So what we've done with NASA is that we have been functionalizing the specific sensors with materials to particular molecules or gases, so volatile compound recipes that we know are very specific to pregnancy. Now, we have more work we can do on that, but what that means is that the sensors that we have on this panel are ones that we have calibrated in a way, or really selected, because they can read and have the sensitivity to the specific gases that we're looking at, that we know from a science point of view, from the GCMS point of view, are what up and down regulate for pregnancy. And we will do the same for disease. So our way of functionalizing the sensors is to the specific molecular recipes that we know, and to be able to ensure that we're selecting the right materials and the right sensor sort of nanostructures that are particularly sensitive to the gases that are important to us.
Thank you. This particular technology, you mostly talked about the pregnancy detection and kind of mentioned the disease detection. Is the disease detection part a functional attribute of the technology as it stands, or is that something you're planning to develop?
So we're in progress with the disease side. So what we've been doing is we've been creating the molecular blueprints, which again is the real fundamental core part of being able to then functionalize the sensors to match that specific, the bias specificity. So to be able for us to do that, we're going through a process that is slightly different than pregnancy trials because we didn't know what we were looking for there. And it was a different journey to now doing biological challenge trials, which we've done once before, where we've looked to go that these molecules are present when the particular bacteria exists, but how does the animal interact with that biologically? And then what is the immunoresponse in particular? So in the past, when we've done bovine respiratory trials, what we've seen is the up and down regulation of the immune system, which is actually really valuable, we've discovered, for those who are particularly dealing with like large numbers of animals in feedlot situations. If they were to be able to identify animals whose immune systems were ramping right up, then they would be able to do something about that. They would separate them out. And then we're working through the process over this next 12 months of being able to functionalize the sensors to particularly identify the particular molecules that relate only, that do not cross over between the bacteria and the viruses. So it's a process, and that's one of the reasons for the fundraising. These things aren't cheap to do. This is really a biotech company applying this to a livestock environment. So the sophistication that we have to go to and the scientific rigor is the same as if it was for humans. That's my approach, because I wouldn't trust anything else. So it is a journey that has to needs to have some additional funding, but the infrastructure is the same. So we've built the infrastructure to be able to do this. We're now working to align the sensor technology.
Thank you. How many tests can the sensor perform before it needs to be recharged?
So at the moment, we haven't hit the limit yet. So it's, the aim is really, it depends how many you do in a day. It's more on the day point of view, so that it's in a cattle environment, if our aim is to do 60 per hour, we have it, the whole goal is to be able to do that for 8 hours a day, because that's what people do. So we're building the product to be able to match the productivity requirement, and we have not hit that. I'm looking at Ash thinking we're just starting to put the new one under pressure, but the other one, it just keeps going. Yeah.
I think, just jumping in here,
Please.
I think the other side of that question that could be relevant to whoever asked it is the longevity of the actual chips. So how many tests can run across these chips? The NASA team, me and Jing, have been using the same chips. They actually pivoted during COVID and used this technology to diagnose, with a small sample size, about 30, I think, from memory. They could predict COVID with about an 80% accuracy. And they've been using those same chips continuously for the last 3 or 4 years, and they're still responding the way they expect them to respond. So that's all part of the learning process. How long do they last? And that's all just part of that, part of that automation process, the printing process, and testing them and coming up with the protocols as we go forward.
Yep. And so what we have done is with the NASA reproduction sensors, which is not the fun looking box, but just behind, it's in this box, which is the chips that Ash was showing you how they were doped. We've now, in the 3 boxes, collected samples of over 1,200 cattle using those sensors. And again, there's no, the sensors themselves are extremely robust. So we'll put that through its pressure test to see whether there is a horizon for that, or when any variability might occur. But we're moving to robotics, and we can do that then in the laboratory because we know the gases that we can continuously put over the sensors.
Great, thank you. Can you explain how exactly the sensor detects analytes
Out of the breath?
I believe so.
Yeah, so I can do the one where do you want to talk about this, because it's where the molecules and actually Mies and Jing are the best ones to explain this about the way the attraction and the repulsion. You do that one of the.
So on each of these little dots, or the same similar sized dots on those rectangular chips, there are what we call doping materials, which is essentially the smarts that NASA's been working on for the last decade or two. and they're made up of nanostructures. So they're carbon nanotubes and other similar types of structures that have different shapes and different, they're called functionalized with different compounds, or they could be some silver in there, there could be titanium in there. Could be other organic compounds associated with those. And as air passes across those sensors, the volatile compounds in the air will react with those structures. And essentially, they'll either donate electrons between the volatile compounds in the gas, or they'll accept electrons, and that will change the resistance across those chips, and the device measures the resistance. So that is how it kind of creates a sense signature. And where that differs, because so when you're using the GCMS, you're actually identifying a particular molecule. It could be a toluene or a benzene or something specific, and you're saying this is the molecule. Whereas there's going to be more than one molecule, because of the way these shapes work, that will react with these sensors. So it's basically a sense signature. And I think the way I think about this is, if you walk into a kitchen and you're standing next to the grill and you're standing next to an oven that's making bread, I could go and take a sample of the gases in those environments and send that off to a GCMS, and it would come back with a recipe of this much gas A, B, C, D, and E for each different one. And if I showed you that recipe of all those gases, you'd have no idea which one was the grill and which one was the bread. But if you smelled that, you recognize that scent signature. And the eNose is all about recognizing those scent signatures through these reactions with the substances, the doping substances on the chips, and then using AI to determine what a set of attributes is, or a set of signatures are, and relating that back to a biological outcome, be that pregnancy or disease, or potentially, a metabolic regulator, or how methanogenic fermentation pattern is.
Good answer, Ash. Don't go too far. Not in more answers like that. One of the other things just to put into there is we are working with other sensor technology that we're commercializing. One is at Macquarie University, and for that we're doing much more closely aligned to biological sensors, the biosensors, where we're using metal oxide frameworks and another range of techniques to exclude any like molecules, because molecules often look structurally very similar. So there are some other techniques that we use, and we may add some of these into the NASA sensor technology as well.
Thank you. I have time for one more question. What are some insights that Agscent has gained from collaborating with NASA that could be beneficial to other companies seeking to commercialize space-related technologies in other sectors?
Okay, this is a really good question, and I think this sort of speaks to the journey. Some of the insights that we've had is that things do take time. As someone explained to me, this is the US government. So to be aware that it's not that it is just going to take time, that to work with NASA and to commercialize technology is coming from a point where they've created it for one particular reason, and to take it to another sector or articulate it in another way actually takes a lot of other bridges that you will need to build. So from us being able to develop the way to collect the sample and that's our own technology, our own IP, is part of the journey of coming together with NASA to do so. It's very much about building relationships. And again, I could not express more the appreciation I have for the NASA staff. I'd also say that to be persistent. No invention comes across either quickly or without hurdles. And if I listed all the things that have gone wrong, as a scientist, I may have come from human science and psychology, but what I've learned along the way is that everything that goes wrong teaches you something. Just because it goes right, you don't always get the same lessons. And so working with NASA has been a part of the lessons is to work through the problems and work through them together, rather than to think, oh, we've got a hurdle, let's just stop. Because NASA, as Ash said, they solve big problems, and their persistence is absolutely astounding. So I think it is, be prepared for it to take time. It will require you to bridge gaps in between the application because it's used for something else. the team and collaboration is really important. But the other thing too is it is sometimes hard. I know for us, because we have an agricultural application, funding is really difficult. It's really tricky. But NASA is someone who to partner with the Reimbursable Space Act agreements, and to be able to work with NASA is a real privilege and a real opportunity. So it's absolutely worth doing.
And just if I can add to that, I think one thing that's very clear is, this is a completely untrodden path. It's not like you can walk into the hardware store and buy any of the ingredients we need to make them. There's specialty places that make them. One chemical that I had to buy, I rang up the company, the website didn't have a link, you had to ring them up, and they had something like a few milligrams left of this one chemical, which was one of the ingredients for one of those sensors. And that's all that they'd had. And NASA's been great in terms of helping to steer us where we can find those things, but it's not like there's a steady path that you're following, and they've been a great partner to take that journey with, because they have supported us. I've spent a lot of time in the lab in the last 12 months and had some, when I've had questions, we've sat down and discussed the questions, and at the end of the day, I think we've both learned from working together, which has been absolutely fabulous.
Thank you both. One last question just snuck in. Have you had experience during your trials with pregnancy loss and watching the breath composition change to open? Also something similar to post-calving?
Yes. So we have actually. So because we're being able to detect at day 18, and there's no equivalent test at the moment, and so we're the earliest, and we actually think we can go earlier. What we have identified is a cohort where, if you think about from a modeling point of view, the tightness is here for those who the pregnancy is secure, and then there are occasionally ones which we would call like borderline outliers, who we would have said actually they were pregnant at day 18, but the loss by 28 or 40, which is not uncommon, is our hypothesis. We've hypothesized that that's actually been the cause of the change, that they've gone from one group to another. And so we can actually track when they sort of move from one group to another. I would like to think that what we've developed here is actually a door opening to a lot more research into this area. Fetal loss, the embryo does not embed in the uterus to day 40. And so early stage fetal loss is a really big phenomena, particularly in the dairy industry, that needs to be managed, and there isn't enough information. So yes, we do see different molecules. We also see from the vaginal probe, which I'm really excited about having more time on. If any of you are on the call and know much about agriculture, you'll know a bull can actually smell a fertile cow probably 2 paddocks away and will do anything to get there. And one of the molecules, a series of molecules we've identified, seems to be the one that switches off when the cow is pregnant. So we know that I love that. This is the beginning of the research journey. We're looking at things with a different microscope, is allowing us to ask some very nuanced and very new questions. So whoever that was that asked, I'd love to talk to you more about that, because it is a really interesting area of research we think there's more opportunity to pursue.
Thank you both. That's all the time we have for questions. I really appreciate you both taking the time to be here today and tell us about this really exciting venture. For those on the call, please do feel free to reach out to Agscent via the contact information on the screen. I hope everybody has a wonderful rest of their day, and thanks for tuning in.
Thanks, everyone.
Thank you very much.